Welcome everyone to the 2021 Life Science XP Education Symposium. My name is Howard Chen, and I am the Education Program Manager at the Biocom Institute. We are excited to bring together educators, nonprofits, and industry partners who are innovating in K-12 life science and biotech education. We would like to first thank our sponsors, Amgen Biotech Experience San Diego, ChromaCode, Elevate Ed Classrooms, and Hologic for supporting our symposium today. We encourage you to visit our sponsor tab on Whova to learn more about these wonderful companies and organizations. For the next 2 days, you will be able to choose from an amazing selection of sessions from our partner schools, nonprofit organizations, and industry who will be sharing the innovations happening in classrooms throughout California. We will have a keynote speaker shortly, fireside chats with industry executives tomorrow, breakout and networking sessions featuring speakers from Bay Area, LA, and San Diego. And the best part, if you have a difficult time choosing which session to attend today, not a problem. We'll be recording every session for you to view after the symposium. Now, before we turn it over to our brilliant keynote speaker, we have a few announcements to share with all of you. The first is a very important inventory of practice survey we facilitate annually with high school teachers. Paulina from our team will be dropping the link in the chat here on Whova. This survey is an opportunity to hear from the amazing life science and biotech educators throughout California. We thank you for taking the time to share about your teaching practices at your school. This valuable information and feedback will help inform how the Biocom Institute can better support a high school teacher community. Next, we would like to invite all our educators to our growing teacher community group on Facebook. Paulina will also be throwing that link on the Whova chat here. The Facebook group is a community space for teachers to share resources and connect with others who are passionate about life science and biotech education. We encourage you to share links, post questions, and comment in this group to help support the community. Now for this symposium, we have to offer here is a certificate of attendance for your school or district if you need to show that for today's event. All you have to do is request it on the event survey at the end of the symposium, and we'll be happy to send you a certificate. And lastly, for only our networking sessions tomorrow from 4:40 p.m. to 5:15 p.m., you will need to use your desktop Whova link, which Paulina will also put in the Whova chat here, where you can participate in these conversations. The reason we need you to do that is the Whova phone and iPad app will not work for switching Zoom breakout rooms. So once again, for tomorrow's networking session, please use the Whova link that is on the chat. And without further ado, I would like to introduce our keynote speaker for our symposium today, Janice Chen. Janice Chen is the co-founder and CTO of Mammoth Biosciences, a startup based in South San Francisco that is harnessing the diversity of nature to power the next generation of CRISPR products. Janice received her PhD from the lab of Professor Jennifer Doudna at University of California, Berkeley, where she developed multiple CRISPR technologies with applications across diagnostics and therapeutics. At Mammoth, she leads the diagnostics team to enable rapid and affordable testing with CRISPR. Ladies and gentlemen, please welcome Janice Chen.
Thank you so much, Howard, for that introduction. I'm going to go ahead and share my screen to start the presentation. All right. I hope everyone can see my presentation. First of all, I'd like to thank the organizers for giving me the opportunity to speak with you all today, and really thank you as the educators for doing the work to help all of us build a better community as we build the next generation of scientists and individuals who are really passionate about life sciences and biotechnology. So I really appreciate all of your commitments to this educational cause as well. So to start things off, I just have a quick outline of where I'll be going with the talk today. First, I wanted to just give a brief overview of my personal background and how I ended up in the CRISPR field, and then also give an introduction to Mammoth Biosciences and our work developing CRISPR-based diagnostics. All right, so I grew up in Salt Lake City, Utah as a first-generation Chinese American, and actually a big part of my childhood was playing chess. My siblings and I competed in tournaments across the country, and when I reflect back on these experiences, they have really instilled critical thinking, hard work, and the courage to sometimes compete as the only young female player in these male-dominated fields. In parallel, my interest in science was also strongly influenced by my dad, who was a PhD researcher at the University of Utah at the time. And growing up, I would visit his lab and learn techniques in molecular biology. And when I think back, having this exposure early on made me fascinated by how fundamental technologies can lead to, fundamental science can lead to really fundamental and impactful technologies. And so when I think about, my early days, I think having this exposure has been incredibly important. I started my undergrad at Johns Hopkins University, and for me, a turning point in my scientific education was actually a course called Build a Genome, which was centered around a mission to build the yeast genome from scratch using short DNA sequences. And so this was really an exciting time for me learning about genomics and how to create basically a minimal viable organism with the power of biotechnology. And this work actually led me to my first independent research in a lab studying yeast metabolism at the School of Public Health. From there, it was a springboard to actually learn how to manipulate genomes of other organisms to produce value-added chemicals like biofuels. And so this was work that I did after my undergraduate career at Harvard Medical School. And during my time there, I got incredible exposure to the field of synthetic biology. And it also made me realize that I wanted to go back and understand how these tools actually work from a mechanistic level. And are there ways that we can enhance them further to invent new and better technologies? So during my time at Harvard, I was fortunate to apply to a number of graduate schools, and I was really attracted to the emphasis on basic science research at the University of California, Berkeley. And so that's where I started my PhD. And at this time, this was 2014, so really near the beginning of the CRISPR revolution. I was just completely fascinated by the technology and was determined to understand more deeply how these enzymes, these CRISPR enzymes actually function. And when I started at Berkeley, I had the fortune to also begin my PhD thesis in the lab of Professor Jennifer Doudna, who, as many of you know, won the Nobel Prize in Chemistry last year for her work developing the Cas9 gene editing tool. So it's been really an incredible journey to get to Berkeley. And on the next slide, I just wanted to highlight some of the incredible collaborators that I've worked with over the years and highlight the fact that science really does take a village. I think one of the remarkable opportunities during graduate schools has been the ability to forge really meaningful collaborations with labs around the world. And some of the names here are just key collaborators that I've worked with throughout graduate school. And I've really just cherished the relationships that I've developed over the shared interest of CRISPR technology. And I think through this process really learned about the power of collaborating with multidisciplinary teams to solve scientific problems. So one of the first multi-lab collaborations that I worked on really focused on investigating exactly how these CRISPR proteins function. So like I said at the time, Cas9 had been really, really exciting and showed a ton of promise as a gene editing tool, but there were still these questions about, how does the protein actually prefer to find its target? What sequences does it like to cut? Can we actually make these enzymes more precise so that they cut their intended sequence while avoiding any unintended cuts. And so that was a real focus on the beginning of my work with CRISPR. And then over the years, I had the fortune of kind of implementing those types of questions onto new CRISPR proteins that were discovered, like this Cas12 enzyme. And through that work, like a lot of scientific discoveries, the finding that these Cas proteins could actually detect DNA was discovered by accident. And so, what started out as basically a biochemical investigation all of a sudden turned into a collaboration to test whether CRISPR could be programmed to detect particular sequences. And so that, in a lot of ways, was really foundational for me in kind of going through the process of discovery to applications. So with the CRISPR-based technologies that we developed in the lab at UC Berkeley, Jennifer, and along with my lab mate Lucas Harrington and I had this shared vision to develop the next generation CRISPR platform. And we really just recognized that there was a unique opportunity to develop these technologies beyond the academic lab, and realized that launching a startup was the first step to further enable these technologies. So after Lucas and I completed our PhDs, we partnered with Trevor Martin from Stanford to build Mammoth Biosciences with a mission to improve lives by reading and writing the code of life. So it's really the programmable nature of CRISPR that makes it a powerful search engine that can be readily modified to target any sequence of interest. So oftentimes we like to think of this as a Google search engine where you can quite literally type in the letters of a disease target or pathogen that you would like to edit or detect, depending on the CRISPR-associated protein or Cas nuclease. And as many of you know, Cas9 is probably the most famous protein, and it has truly paved the way for genome editing. And like I mentioned, over the last several years, there just has been an explosion of new CRISPR systems that have also been discovered and characterized. And a lot of these novel Cas proteins have unique properties that enable an expanding toolbox for CRISPR-based applications. And these different applications include things like detection, genome editing, of course, and things like genome modulation that gives you really exquisite control over developing genomic tools. So, one really important part of CRISPR is that in nature, CRISPR is actually an ancient immune system that bacteria use to fight against viruses. And so by surveying diverse microbial environments, such as hot springs or lakes or soils that we indicate over here on the left with the funnel, we can actually discover and mine tens of thousands of novel CRISPR systems that just exist in nature. And we can actually bring those back into the laboratory to start understanding the capabilities, basically within a test tube. So at Mammoth, we take this discovery first approach to uncover new CRISPR systems. And basically by harnessing the diversity of nature, we're able to leverage this growing toolbox of Cas proteins with unique capabilities that will enable solutions in life science research, in healthcare, agriculture, biodefense, and much more. So it really is a really incredible process that nature has evolved over billions of years that we can leverage to create some really groundbreaking technologies. So our core areas at Mammoth focuses on discovering new Cas proteins, as I've described, including the Cas14, Cas-B, Cas13, and Cas12 protein families that together are capable of advancing disease detection and treatment. So CRISPR really is an extraordinary technology platform with the potential to deliver on the promise of precision medicine across diagnostics and therapeutics. For the remainder of the talk, I'll focus on how Mammoth has been harnessing the power of CRISPR as a programmable diagnostics platform. And this is one that uses rapid enzymatic recognition to achieve high molecular accuracy while maintaining the ease of use to detect virtually any genetic sequence of interest. So just a quick overview on some of the diagnostic capabilities of our CRISPR proteins. We can repurpose CRISPR, as I said, as a search engine that can find and detect any nucleic acid target, and we call this technology detector. And the way it works is that once that Cas complex recognizes a matching target, the Cas protein activates this non-specific shredding activity, which is capable of cleaving these reporter molecules that can provide a diagnostic result. And the distinct families of CRISPR proteins that have this capability include the Cas12, Cas13, and Cas14 enzymes that naturally target either DNA or RNA sequences. And so this really enables a wide flexibility when you're designing diagnostic assays. In the next slide, I'll actually show a little video in terms of how this chemistry works within a test tube. So again, just to clarify this further, we pair the Cas protein with a guide RNA that's programmed to match the exact sequence we want to detect. So these molecules form a complex that searches through billions of letters and cuts that matching sequence. But like I said, once these proteins find that target, they become this non-specific shredder, and they're able to cut apart these molecules that create a color change that only happens when that target sequence is present. And so in this way, you can get a very specific detection using this CRISPR protein. So the flexibility and simplicity of the detector platform allows it to be used across a continuum of testing solutions. So on the left hand, a single-use disposable molecular test has the potential to place the accuracy of a molecular lab into the hands of consumers. And this enables decentralized testing to detect diseases earlier and in the most vulnerable environments. Diagnostic testing in centralized labs on the right are also needed to process large sample volumes with high performance. And we think about workflow improvements that will enable even greater testing throughput. And so CRISPR is one of those unique technologies that has the potential to advance technologies across this testing spectrum. So when the COVID-19 pandemic hit in early 2020, we recognized that our detector platform could be rapidly reconfigured to detect SARS-CoV-2. So what I'm showing here is just a timeline of some of the key milestones and events that we worked towards as far as being able to take our technology platform, which is primarily in the R&D phase, to actually be able to scale that and deploy it as a diagnostic test. And a lot of this work started early in 2020, and our first big moment was our peer-reviewed publication in Nature Biotechnology, which was the first CRISPR-based method to be published for the detection of SARS-CoV-2. And from there, we were able to take that work in collaboration with Charles Chu at UCSF to deliver on for FDA EUA submission, and we received the authorization in August of last year. In May, we very quickly moved forward with GSK on a partnership to develop a CRISPR-based consumer diagnostic test for SARS-CoV-2 because of the importance of being able to take diagnostic testing closer to the consumer when it comes to a pandemic and being able to detect diseases in decentralized locations. In July, we were fortunate to also be the first CRISPR company to win an NIH RADx contract to commercialize our high throughput SARS-CoV-2 detector boost platform, which I'll talk about in a little more detail in the upcoming slides. And for us to achieve that level of scale up and manufacturing, we also had the fortune of partnering with companies like MilliporeSigma, who we work with to manufacture our chemistries that we develop at Mammoth. And we've also been able to partner with automation companies like Hamilton and Agilent to actually implement this full turnkey workstation. Later in the year, we also were fortunate to receive support from DARPA to advance next generation CRISPR-based diagnostics for biosurveillance platforms. And this is ongoing work that will deliver on the future of CRISPR diagnostics as well. So it's been a really crazy year for all of us, and we've all been impacted in a lot of different ways. And I think it's been an extraordinary year as well for synthetic biology and biotechnology to be able to be part of the solution. So just to give you a quick overview on the high throughput detector boost SARS-CoV-2 platform that we have been working on in collaboration with our NIH RADx colleagues, I just wanted to provide a kind of overview of how that system works. So when we think about centralized environments, such as labs that are limited by capacity, Mammoth has developed a turnkey workstation that runs a CRISPR-based SARS-CoV-2 molecular assay that is based on automated liquid handling robots. So the lab worker simply loads reagents and samples onto the robot, presses a button, and simply walks away. And the robot is capable of executing the automated workflow that includes sample extraction, target amplification, and then CRISPR-based detection. And this high throughput system can perform up to 1,500 tests within a single work shift. And this is possible because of the minimal requirements to run the assay. In addition to the benefits of the increased testing capacity with less hands-on time, our detector boost SARS-CoV-2 reagent kit also has performance that rivals RT-PCR. It's compatible with nasal swabs and saliva sample types. And because we don't depend on the traditional PCR-based reagents, there is also, it's less prone to supply chain risk. And again, with the support from the RADx program, we've been working with Millipore Sigma to manufacture the commercial lots of our detector boost assay, and working very closely with our automation partners at Agilent and Hamilton to develop this complete solution for laboratories that are looking to scale up capacity. And so by implementing this sample to answer solution, we hope that CLIA labs will significantly improve the capability to test large groups of symptomatic and asymptomatic people. And so we believe that this will help increase SARS-CoV-2 testing capacity and provide the necessary safety and security to address critical aspects of the economy. So this has been an effort that is approaching a commercial launch in the near future. So I just wanted to also highlight that, whereas traditional clinical diagnostics require infrastructure within a centralized laboratory, which is where our detector boost platform fits within, there's also, the future of CRISPR Diagnostics, which is what we're working towards, and being able to deliver on something that is engineered to have the accuracy of molecular diagnostics in an ultra portable, low cost format. So our ultimate goal is to deploy CRISPR to power diagnostics anywhere and everywhere. And we believe that the future of diagnostics will enable a world in which all patients have access to high quality diagnostics that can be administered in the privacy of your own home to inform your personal health status, and followed with an actionable treatment plan. So this is something that we believe that CRISPR has the potential to deliver on. And finally, I just wanted to provide a snapshot of the growth of our team from our humble beginnings in a biotech incubator in the Dogpatch neighborhood of San Francisco, which we very quickly grew out of within our first year of operation. We found our next home in South San Francisco on Verily campus. And this is actually where we operated during most of the pandemic. And just a few months ago, we've moved the company to our new headquarters in Brisbane, which is where we now call home. And so we've grown from, a group of, 10 people in our incubator to now occupying quite a big, quite amount of space in our Brisbane location with just over 84 full-time employees. So it's been really incredible growth in the last couple of years as we deliver on some of these opportunities. And so with that, I just would like to extend a tremendous thank you to the entire Mammoth Biosciences team and our partners and funding sources for all of their contributions throughout this journey. We could not have been able to get here without the full support of everyone who's been a part of this mission. And so I would also like to thank this audience for your attention, and I'm happy to take any questions.
Hi, Janice. Yes, we do have a question. And the question is, this technology is so incredible. In some ways, it feels like the sky is the limit with its application. Can you talk about your greatest hopes for the technology, as well as your biggest concern regarding ethics and how it will be used?
Yeah, I think it's a really important question in terms of understanding the potential of the technology and also the responsibility that we have as far as being able to deploy. I think for me, my greatest hope is that we'll be able to create these tools that have really incredible genomic precision that allows us to do everything from sort of like genome surgery to address some of these really devastating diseases that we understand the genetic basis of, all the way to being able to detect, these diseases and being able to use those to help us even create better treatments. And so I think we always have to ask ourselves, what are the ethics of actually being able to use these gene editing technologies? But certainly a lot of the diseases that we've been focused on are, areas where there is, a known kind of genetic cause for that particular disease, and being able to use that as a potential treatment where there are no other possibilities. So I think that's an area where, we want to be focusing the technology for good as far as being able to treat the underlying cause of some of these genetic diseases.
Thank you.
So while we're requesting more questions, you feel free to go on your Whova app and type in your questions on Q&A. I do have another question here, Janice. What is one piece of advice you would give to young students who look up to you and are interested in pursuing a future in biology?
Yeah, I think one thing that I've always told people is to make sure that they find their passions and kind of think about what is their North Star as far as what they really want to achieve. And I think if you can find that North Star and look for, ways to get there and find the support, I think that's been really important for me, and also what I can help as well in terms of helping people, get the mentorship that they need to understand how to get there. But I think at the end of the day, understanding what those passions are and not giving up on them is part of the journey. And yeah, I think the other part of it is to also have fun, because it's all, the present can move so quickly, and that's something that we've all learned in the last year as well.
Thank you. And we do have one more question. Is cancer one of the diseases that could be diagnosed by CRISPR technology?
Yeah, so a lot of cancers have a genetic basis, and we understand that there are certain mutations that drive those diseases. And so from that perspective, if we can understand that origin of the disease, you could envision that CRISPR as a gene editing tool would be able to be part of that therapeutic solution. So certainly cancer and applications in immuno-oncology is one active area of research using CRISPR technology as a potential therapeutic.
Thank you, Janice. I have a couple more questions here. Has CRISPR played a role in development of COVID-19 treatments or vaccines?
Yeah, there's a lot of research ongoing in the area of COVID-19 treatment, in the sense that you could potentially use some of these CRISPR enzymes to potentially identify and even destroy the SARS-CoV-2 virus within a cell. I mean, this is certainly in the early stages of research, but some interesting proof of concept has been demonstrated. I think there's other exciting areas where people have used CRISPR technologies to kind of probe the status of the cell when it goes through different stages of disease progression for COVID-19, and understanding and identifying potential therapeutic targets using CRISPR as a tool. So there have been a lot of different creative ways that people have been using, this search capability of CRISPR to produce better targets for vaccines or therapeutics.
Thank you. Next question we have here is, are there tools available for educators to teach CRISPR in the high school laboratory?
Yeah, one of the great tools that I've used is from the Institute IGI at UC Berkeley, and they have really nice educational tools that describe everything from appendix describing some of the vocabulary that I've been using. What is a Cas enzyme? What does CRISPR stand for? So the IGI that is also founded by Jennifer Doudna has a really great set of educational tools that I encourage everyone to look into as well.
Alrighty, and we do have one more question. Does Mammoth Bioscience provide paid or not summer internships for high schoolers?
That's a great question. What I can share right now is that we are planning to put together such a formal internship program at Mammoth, hopefully launching by next year. So at the moment, we don't have a formal program, but we're always open to any opportunities, and love hearing from people who are interested. And we can always take a look, of course, on a case-by-case scenario. But we are driving towards launching a paid internship program next year.
Thank you. And one more question. What would you emphasize about CRISPR if you taught students in high school or college about the technology?
Yeah, I think I would emphasize that CRISPR has revolutionized the way that we can interact with our genes, and whether that looks anywhere from being able to detect genes from pathogens all the way to editing genes within our bodies. So I think, again, having that precision all the way down to the nucleic acid level has really opened up a number of opportunities that hasn't been so readily possible with existing technologies. So at the end of the day, I think the search engine concept is really powerful when you try to explain how CRISPR works, because it's, at the end of the day, a programmable system, and that RNA molecule is what guides CRISPR to actually perform all of these functions that allow new kinds of diagnostics and therapeutics.
Thank you. And can you please share the range of academic backgrounds and experiences of some of your team members?
Yeah, so our team members have a hugely diverse, I would say, background from skill sets. We have people ranging from, just having their a bachelor's degree all the way to people who, have received their PhDs, postdocs, have been in industry for, decades. So it really does span the gamut. When we've had interns in the past as well who have, were in their undergraduate career. And so they've been able to, get into the lab and actually help run experiments as well. So I would say there's certainly quite a range of expertise that we've had at the company, and I think part of what makes a startup really exciting is that it's a small, tight-knit group, and everyone is has the opportunity to get a lot of exposure to a number of different types of expertise.
Thank you. And I think we have time for just one last question. What is the best approach to contact Mammoth regarding partnerships?
Yeah, I think that I believe info@mammoth.biosci is an email that it should be on our website, which is mammoth.bio. And so if you just use the contact us page, we do have people monitoring that. So feel free to visit our website, and that would be a great way of contacting us about questions regarding partnerships and anything else.
Thank you, Janice, for sharing your time and expertise with us today. We learned a lot about CRISPR technologies, and we look forward to following you and your company in their future developments.
Thank you so much for the opportunity and really excellent questions.
At this time, everyone, we are encouraging you to take a quick break here and then jump back on at 3:40 p.m. We have 3 wonderful breakout sessions. One is featuring Illumina, The Future is Bright, bringing genomics and DNA day into your classroom. The second is Kristen Wolsogel and Yingsu Liu, who are going to be from Baybeck, who are going to be talking about collaborative resource sharing and supply chain models for biotech education. And then the third session is Helix Charter High School, building a biotechnology pathway at the high school level. So we'll see everyone in their breakout rooms at 3:40. Go look into your Whova app, and you'll be able to access those sessions there. See you guys there. Thank you for participating in our keynote.